And so I'm going to introduce John Zaia. He's chair of the City of Hope's Department of Virology and is holder of the Aaron D. and Edith Miller Chair for Gene Therapy at the Beckman Research Institute at the City of Hope. John has a phenomenal career in clinical virology research. He came to the City of Hope as Director of Virology and Infectious Diseases, and since 1999 has been Chairman of the Department of Virology at the Beckman Research Center at City of Hope. He's directed the Clinical Research Center satellite at USC School of Medicine, has been Chairman of the City of Hope IRB and of the Clinical Research Governance Board, and is currently Deputy Director of the City of Hope Cancer Center for Clinical Research. He's authored over 180 published papers, holds 10 patents, has NIH funding, and served on numerous review boards and is currently a member of the NIH Recombinant DNA Advisory Committee. So, Dr. Zaia, and after John talks, he'll introduce Loring Leeds, survivor of AIDS lymphoma, another patient living with HIV, and has already participated in a gene therapy trial for people with HIV. Thanks, Jeff.
Thank you, Jeff. And thank you to the CIRM board, to Bob, to Alan, to all the members of the staff that are here and who have been very helpful to us in our application for this funding. It's my pleasure to describe the project to you today and to put it into perspective. So the statement of the problem was most eloquently articulated by Jeff just now. Basically, the problem is that with the best treatment we have today, virus infection continues. If you were to measure people who have undetectable virus, that is, it's undetectable by conventional assays, that is, the assays you can get from the usual laboratories, it's undetectable. But if you go to a research lab where you have what's called single copy assays, you can always find HIV circulating in the blood. And that's what Jeff's referring to, that virus is undoubtedly doing some damage and it's going to ultimately have some problems over a lifetime. Where does this virus come from? Well, we say philosophically that it comes from a reservoir. We don't know where that reservoir is, but it must be a collection of cells that are protected in some way from the treatment. That is, that the drugs cannot get to some parts of the body, and it's in that reservoir that the virus can grow. We don't know that for a fact, but that's the theory. So the question is, how could you then treat the patient optimally? Well, you must then attack that reservoir, and if you can do that, you could at least eliminate the duration of therapy, and it's possible that you may be able to eliminate the need for treatment completely. And that's our goal. So if you look at it graphically, this is what Jeff was referring to here. You have in blue here the effect on the immune system over time. So this is time, and this is your CD4 count. So over time, the virus is in red. It infects you, and it's a peak, and then it settles down to some level that's think of as a virostat. That is, each person has their own level, and they tend to maintain it. It may be 200 copies, and maybe 50 copies, and maybe 10 copies of virus. But it continues for many years, and we tend not to treat until you reach a certain drop in your CD4 count, usually between 300 and 400. That's a critically controversial point, and Jeff is very involved in that in San Francisco, where the decision has been made to treat as early on as possible after infection. And whether that will be accepted by the public remains to be seen. In any event, this is the window of opportunity for some genetic manipulation. And if we could, in fact, introduce cells that are protected, then we could minimize the amount of virus and keep it at that low level and prevent it from affecting the T cells. One of these is supposed to do that, but it doesn't. Okay, so how would we do that? Well, this shows you the cells that are generated from the bone marrow. And it's generated from a multilineage stem cell. And this is the mother cell of all of our blood cells. All kinds of cells, but primarily the cells that HIV grows in are the T cells and the monocytes. And theoretically, if we could introduce a gene that is protective here, you could then develop T cells and monocytes that are protected. All right, so how do we propose to do that? And we propose a technology that is, let's say, native to California. It was developed in California at a company called Sangamo. And it's being utilized by investigators at USC. Paula Cannon, who is here. Paula? Paula's right here. And David Augusto leads our team at City of Hope. And the question is, how do you get zinc finger nuclease into a stem cell? Well, the question really is, what is zinc finger nuclease in the first place? Thank you very much. Well, without going into a lot of the science of it, it's a molecule that has been engineered to attach to DNA with so-called fingers. And these fingers are small peptides that were discovered in proteins that were already known to bind to DNA, transcription factors and things that need to interact with your genes to get them to become expressed. So these were taken and they were combined with again, a natural enzyme that can cut DNA. And together, the fingers then attach to the site of the DNA, and the enzyme is right there, and it then breaks the DNA. It makes a double-strand break. So this is the DNA, this is the binding proteins, and this is the enzyme is called FOC1. So it contains these two components. It's a designed molecule. It could really be engineered to find any abnormal gene and either remove it or correct it. So, for example, it could be targeted to sickle cell anemia or some other gene. In fact, this is one reason why we think our work here will have great applications to other interests of the CIRM-related projects. The natural repair process can either then destroy the gene, or you could introduce a corrective element that could be inserted in here. And so, in fact, insertions can be made that are the correct gene. For our purposes, we just want to remove a gene that is important for AIDS virus. And what is that? Well, this is called CCR5. This is the attachment that is important for the virus to get into a cell. You know, I tend to think of it as kind of like the doorknob of the cell. So if the virus can attach to this, then it can enter the cell. It's one of the major co-receptors, it's called, of the virus. An absence of this protein tends to protect a person from getting HIV and AIDS. And it has become a validated target for drug development, and there is already a drug available that uses this principle and inhibits CCR5. Okay, let's look at this just for a second. This is the gene shown graphically. It's a long gene, and it goes 7 times through the cell membrane, which is shown in blue. So this is a cell surface protein, and the natural mutation is a deletion right here that alters the rest of the chain and prevents it from being present on the cell surface. This is not a rare mutation. It's found in Northern Europe, as shown in this slide here, and it's a very interesting reason for that. And that is the protein itself is important for inflammation. It's not critical. That is, people who have it live a perfectly natural life. In fact, I mean, people who don't have it, but they don't get arthritis, and they don't have as many myocardial infarctions. They have less inflammation. If they get attacked by West Nile virus, they have more problems than you and I do, presumably because of this. And why it's in Northern Europe is thought to be due to the fact that this is where the bubonic plague was present and removed, a third or more of the population back in the Middle Ages. Those who survived had less disease, less buboes. These buboes are these large hemorrhagic lymph nodes that got infected and brought on death. So if you couldn't make the big hemorrhagic buboes, you survived. And you carried on to your children, then, this mutation. Well, Jeff referred to a patient who got transplanted in Berlin, the Berlin case. And he had the good fortune of developing leukemia in Europe. And he also had AIDS. So his doctor said, I'm going to look for a donor, not just that is matched with him, but a donor who comes from an area where we can find a mutant, a delta 32 mutation of the CCR5 gene. And that's exactly what was done. I just want to say that if you're homozygous, that means you have 2 copies of the gene, you are resistant to HIV infection. And if you have 1 copy, you just have delayed progression. So this patient underwent the transplant. Now this patient actually was a citizen of the US from Washington, D.C., but was working in Germany. So Gero Hutter is the young physician who told the staff, let's wait. I've read about the CCR5 issue, let's find a donor. He did, and the patient then engrafted. The leukemia went away, but with it, HIV went away. He went off drugs. He's now out about 4 years. He's been off therapy for all except for a few weeks of the immediate period after the transplant. So since that time, after transplant, he's been on no therapy. He has had multiple attempts to find virus. He's had biopsies of rectal mucosa, biopsies of brain, blood analyses, single copy assays to detect low levels of virus, and nobody can find virus in this patient. So this is a case that rejuvenated the field, and this is what got some of us much more excited about looking for methods of applying this to the general population. Essentially what we're trying to do is make a donor, such as this patient found, that is CCR5 deficient. So we want to basically apply zinc finger nuclease to the multilineage stem cell and create a defective progeny that will be CCR5 negative. Okay, so how are we going to do that? This shows it kind of simply. That is, we're going to put the gene into a virus. It's an adenovirus, shown here. We're going to then infect stem cells from patients. We're going to transplant them into the patient. We're going to allow then the cells to engraft, and we expect that HIV will kill off the non-genetically modified cells. And so only the genetically modified cells will continue to grow. And as these cells grow in the patient, the HIV will be controlled. Now that is a dream, but I'm going to show you some data from Paula Cannon, who has reproduced this in a mouse. And we think that because we can do it in the best animal model we have, that this has a theoretical likelihood of occurring in patients. So our goal in the CIRM project is to get to this point of where the patients are actually treated. Okay. Now, our work is basically to answer the questions that the FDA, the Food and Drug Administration, wants to know. Well, what do they want to know? Well, they want to know whether we can explain the treatment and we can explain the manufacturing process for their satisfaction. We think we can do that. We have expertise here, especially from David Augusto, who has already done it with other projects of ours. You're going to meet a patient in a minute who has actually received some of those cells that came through our process. So we believe we can explain this very nicely, and that is much of the work that we have to actually scale up from a mouse-sized treatment to a human-sized treatment. We have to show that the treatment then at that scale actually would work in the mouse as we've seen it to work when we're working at small scale. That is, does it knock out the CCR5 gene? Are these stem cells still functional? That is, can they still make the progeny that we want them to make? And in the process of cutting that gene, did we cut other genes that might lead to cancer? Is it carcinogenic? I mean, when you alter the human genome, or any genome, you run the risk of changing events and producing the potential for development of cancer. So the carcinogenicity studies will be key, and they will take a considerable time to complete because they take time just to watch the treated animals. Can we get approval from the institutional and the federal ethics committees? The NIH Recombinant DNA Advisory Committee is one, of course the FDA is the other, and then our own institutional IRB and the IBC. So that's really the work of this proposal. Now, what is the evidence that we can actually pull this off? Well, I've alluded to Paula Cannon's work at USC, this test of concept. she has a mouse model in which the mouse is, immunodeficient. It's called an NOD SCID gamma C knockout mouse. That means it's lacking a number of immune factors that allow it to eliminate foreign cells. And so we can put in a foreign cell, a human stem cell, and they will grow in this mouse. The mouse has to be irradiated first, but the mouse will develop a very nice human immune system. It'll have cells in its thymus, it'll have some cells in its lymph nodes, and those cells are human cells. So you successfully engraft them, and then you can treat that mouse with an HIV infection and ask the question, what happens? And did this zinc finger treatment help it or not? Okay, so this is a little complicated, but let me just take you through it for those who are not used to this. This shows the types of cells that we're looking for. This is a T cell, a CD3 cell. This is an important lymphocyte. This is a CD4 cell. This is a CD4 cell, and this is another kind of cell here, is CD8. So if you're high here, you have a lot of CD8s, and if you move in this direction, you have a lot of CD4s. And there's 3 panels, a control, and then a control with or without zinc finger treatment with HIV infection. So he took these are lymphocytes. Each of the little dots are lymphocytes. So they're from the gut mucosa, from the spleen, or from the thymus. And you can see that they look different. And basically what you're seeing here is that when we treated when we did not treat the mouse with zinc finger cells, but gave him HIV, the HIV killed these cells. These cells then were the CD4 cells, and well, it looks like the CD8 cells as well. The jointly positive CD4 and CD8 positive cells. But with the zinc finger, these cells remain robust. And this was a remarkable finding, and it was really a proof of concept in the animal model. So what actually do the cells look like? Here there's actually stained with a brown stain for CD3. This is the T cell marker. And this is the lymph node from the gut, and you can see the brown staining here in the lymph nodes. And the brown stain remains in the zinc finger population, but not in those mice that did not get the zinc finger treatment but were HIV infected. In addition, these mice were looked at for their CD4 counts. And this is shown here in terms of the counts and the weeks after treatment. And those that were the controls went down rapidly, and all the cells were destroyed. Whereas the CD4 counts went down briefly, but then something happened. And what was happening is the protected cells began to predominate and to grow, and they grew back to the normal level. And this was the surprising finding, that the HIV seemed to actually select, and that was the key to, I think, Paula's discovery, which we hope will be published shortly. If you look here, there's 2 panels. Let's look at the left panel first. This is a test that determines what the gene looks like. So here's the gene for CCR5, and it's shown from multiple tissues. That's the, for example, the bone marrow, the spleen, the liver, the lung, the thymus. And these are the different parts of the intestines, the large intestine and the small intestine, the epithelial cells, et cetera, et cetera. So this is the gene, and this is the gene in the zinc finger untreated, and this is the zinc finger plus the HIV. So zinc finger uninfected. So the zinc finger cells have a small percentage of cells that actually have the cut of the CCR5. So we cut the gene now, and you can see that in a small percentage it is now in two different pieces. And it's shown here that is in black is the percentage of the CCR5 genes that are disrupted. Not that many, but about 5 to 10% from each of the tissues. This is in the uninfected animals. So that when we engraft these animals after transplant, we have 5 to 10% of the circulating cells, or the cells in these tissues that are, quote unquote, genetically modified. But after infection, it's a much larger number. In fact, some of these, it reaches 80% of the total tissue reservoir of these cells. So the HIV has allowed selection of the protected cells. And that's the critical question that we're going to be asking in the clinical trial. Will the HIV infection that continuously occurs in these patients allow selection of these cells over time? Now, the other thing that's very interesting is Paula followed these animals for several weeks and did the kind of assays we do on patients, that is a PCR assay for the amount of virus that is present. You can see the amount of virus that's present is quite high in the control. It falls off with time as the animal begins to deteriorate and the cells are lost. But the treated animals, shown in the white, eventually reach a point by 10 to 12 weeks in which no HIV can be detected. So this is a reproduction of the Berlin case. That is, we cannot, by the best tests we have, find HIV in these animals. Now, that's a remarkable thing to imagine that could occur, but that's certainly what is being seen in this situation. And part of our challenge will be to see whether taking the kinds of cells we have access to, namely cells that are used in a bone marrow transplant for a lymphoma patient, will these cells be able to produce the same effect? What Paula uses for this is a different kind of cell. It's a cell that comes from a placenta. It's a cord blood cell, which is a much more robust type of stem cell. And our patients have been through a lot of chemotherapy, and their stem cells have been battered. And it's remarkable that they do as well as they do. They certainly are therapeutic, and they engraft in patients. But whether they'll perform as well in a foreign situation like this remains to be determined. Okay, now what CD4 contributes to this is expertise in how to do bone marrow transplantation. That is, how to get these cells out of the test tube and into the patient. And for the past 15 years, we've developed a platform for testing genetic therapy, if you want to look at it from the standpoint of the scientist, but from the standpoint of the patient, we've developed a way to treat AIDS lymphoma when nobody else would treat AIDS lymphoma. And insurance companies would not go near these patients. We established this method and argued in San Francisco for payment for these cases, and now it's a standard of therapy for the treatment of the AIDS lymphoma patient who has relapsed from his conventional therapy. Conventional therapy is to give chemotherapy, and many patients do well with that. But if you don't, the only thing that's going to cure you is a transplant, an autologous transplant. So we collect stem cells. We collect them by giving a medicine called GCSF, standard treatment. We collect it and we purify the cells. Then we genetically engineer, for this experiment we do now, we genetically engineer these cells and give them back to the patient. And we propose to hold the backup cells that are untreated and give those if needed. But the first experiment we did was, and during this time, the patient receives this ablative therapy, we call it. That is high-dose chemotherapy to destroy the lymphoma. In the destruction of the lymphoma, we destroy the marrow. And this is a lethal treatment if you don't rescue with these stem cells. So the stem cells are given, the patient then recovers in about 10 to 20 days. And this result has been excellent. That is, about 80% of these patients are in long-term cure for 5 years, 10 years, and you'll see Lauren leads in more years than that. In our actual proposal to the FDA, we're going to propose not to infuse these cells back. But the very first experiment we did, we infused these cells back because we felt that the therapeutic obligation was primary. The ethical demands were that we treat this patient with this potentially curative treatment. However, this is actually the result of that study. This is a study of David Augusto's, in which you're seeing here the percentage of cells in the peripheral blood over time. So we treated 4 patients, and we followed them for almost 2 years now. And you can see that some patients went up high early, and some went up higher levels later. That is about 16 months. But these levels are not, relatively speaking, these are not that high. they're less than 1% of the total cells. In fact, they're less than 0.25% of the total cells. And the reason for that is that we overwhelmed the system with these backup cells. Excuse me. So the ratio of these backup cells to the other cells was about 0.15%. So that's what we learned, that in the future we want to maximize the number of cells that we deliver. Now, the other thing that we've learned is that two of these patients that have the highest levels had some breakthrough HIV. And we think that that may have stimulated the outgrowth. That is, this patient had HIV very early. This patient here had HIV very late, around 15 months. In fact, the patient himself, against our advice, went on a drug holiday for 3 months. And following that drug holiday, he was down at this level. At 12 months, following the drug holiday, he's gone up. So we think that, in fact, even with this genetic mechanism, which is a different one from the zinc finger, there could be selection by HIV. So this is the team right here. You met Paula. You haven't met David Augusto. David, would you please raise your hand? John Rossi is our basic scientist on this team. I don't think John is here today. Larry Kuchar is involved in the regulatory affairs and the good clinical practice advice for this project. So we want to develop an FDA-compliant process for treating human stem cells. We want to scale up from the mouse size to a human-sized product, demonstrate that these cells still work and are safe, and we want to complete the final application to the FDA for approval to begin the clinical trial. So that's basically the project. In summary, I think that there is a need, I hope I've convinced you there's a need for improved treatment of HIV/AIDS, that the zinc finger technology offers promise for stem cell-based improvement in the control of HIV. In fact, I think it offers a lot to CIRM in general for applications that are going to be needed in the future, for knocking out genes that may be critical for control of, let's say, malignancy, the potential of these embryonic stem cells. Our team, we think, has experience to bring this project to the clinical test. And as I say, the information learned here will have application to other methods of zinc finger-based stem cell therapy. So thank you very much.